Hello, welcome to this guided presentation. My name is Silvia Bonilla and I work as a scientist at MIMETAS. And here we are going to show you a poster that it's about one of the models that we built in our organoplate Graft technology, which is placing a 3D tumor model that can serve as a platform for drug screenings or endothelial and tumor cell interaction studies. So the poster is titled A High-Throughput Microfluidic Platform for Vasculature and Ovarian Tumor Blood Co-cultures. And what we did here overall was to place a piece of microtissue on top of a vasculature bed and then expose this co-culture to different drugs. I'll explain every figure step by step. So first, I want to introduce you to the organoplate graft, which we have here on the top of the poster. The organoplate graft is a platform that is based on a 384 well plate format. And in this plate, we have multiple chips. And in each chip, you can build a mini 3D tissue model. In this case, it will be a mini tumor model. So the advantage of having a high throughput system is that you can have multiple conditions in the same plate, such as different cultures with multiple cell types, or for example, different drug exposures. But something that characterizes as well the cultures in the organoplate is that they are membrane-free. So you have different compartments in each chip without the need of any membrane. And the cultures are also perfused, which means that we place a plate on top of this gadget that we see in this figure, which is called the organoflow. And this organoflow will shift from one side to another, helping the medium to go from one of the wells to the other side of the microfluidic channel. And thus, it will create a bidirectional flow that will be beneficial for the cells to have nutrient flow and oxygen flow. So how do we build the models in the organoplates? Then it brings me to the next figure. And it consists in first adding a liquefied extracellular matrix. So we load the extracellular matrix, which is liquid, and thanks to the presence of this phase guide technology, it doesn't allow the gel to jump into the perfusion channels, which are these two red lanes through here. And once the ECM is formed, we will see the endothelial cells on the sides. Once we have the endothelial cells there, we will place the plate on top of the organoflow. And just by having perfusion, the endothelial cells will proliferate and form 3D tubes. The next step is to form a vascular bed, and we do this by just adding an angiogenic cocktail in the center of the gel chamber, and this will create a gradient that will help the endothelial cells to form tip cells and form sprouts that will develop all the way towards the center and form eventually a vascular bed. Once we have the vasculature bed, we will add a piece of tumor tissue organoid or spheroid that we want to add, and wait a couple of days so that it can interact with the vasculature bed. And after that, you can just study the way that they interact, or you can, for example, add a third cell type, for example, T cells, or you can add a drug, which is our case here. So at the bottom of the schematics, you can actually see day by day how the phase contrast images look like. The endothelial cells here are marked with, and are labeled with RFP proteins, and they are also red. And then we see the tissue and how over time there is some interaction and there is things happening in this microchip. So one of the assays that you can perform is just doing immunostaining to check for the expression of specific markers. This is very easy, and you can have very high quality images in the organoplate because on the bottom of the chips there is a glass which is very thin and allows high quality imaging with confocal microscopes. But what we can see in this image is, for example, that we stain for CD31, which is a injection marker of the endothelial cells, and MUC16 in red, which is expressed by the ovarian tumor that we have in culture. So in the next figure, we can observe the results that we obtained after exposing the microtissues to different drug treatments. And we had control conditions, palbociclib, paclitaxel, and sorafenib exposure. So what we wanted to quantify in this drug exposure assay was the proliferation rates of the different cultures in these different conditions. And what you can see in red is the EDU, which is a marker of proliferation. So the more red signal we saw in the cultures, it meant that the tissues were proliferating more as compared to the other ones with less red signal. So if we put it into a graph, we can see that in control conditions, there was overall more proliferation as compared to the treated conditions. Especially sorafenib, the anti-angiogenic compound, really prevented the tissue and the endothelial cells to proliferate anymore. And overall, something that we can also observe is that the proliferation in co-culture conditions, what we see in orange, was normally higher as compared to the monoculture conditions, which means that the tumors tend to proliferate more when they are together with endothelial cells. This doesn't happen in sorafenib because, again, this is an anti-angiogenic compound, and the vasculature bed was actually really damaged. So the take-home message here will be that these cancer-on-a-chip models are suitable to recapitulate the tumor microenvironment, mostly if you use a piece of microtissue or a piece of biopsy, but you can use organoids or spheroids here, and then use these kind of co-cultures to study tumor disease or progression, to study tumor or endothelial cell crosstalk, or also do tumor microenvironment studies. And as this platform is also a high throughput, you can do drug screening studies of anti-angiogenic compounds or anti-cancer compounds, and especially be able to design or test and predict which drug will be more effective for a specific patient-derived material or a specific cancer type. Thank you very much for listening. If you have any questions, just don't hesitate to contact us or visit our website. Bye.